Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com Inside Scientific and Indigo Biosciences webinar titled Save Time and Money When Prioritizing Drug Candidates Using Cell-Based Reporter Assays for Nuclear Receptor Profiling. I'm Liam Samuel from the events team here at Scientist.com, and I'll be the host. So today we're being joined by Dr. Jack Vanden Heuvel, Professor of Veterinary and Biomedical Sciences at Penn State University, and co-founder and CSO of Indigo Biosciences. In his presentation, Dr. Vanden Heuvel will discuss the use of cell-based reporter assays for nuclear receptor profiling to efficiently prioritize drug candidates and minimize off-target effects.
All right, thank you, Liam. And what I'd like to do today is go over what's shown here in the outline, talk a little bit about the drug discovery strategy, a very brief overview of nuclear receptors and nuclear receptor assays. And then I'm going to talk about how you can use nuclear receptor screening and profiling to try to prioritize candidates within a drug discovery strategy. So let's start out with discussing the current problems with drug discovery and the strategy that is being used. And I'm sure many of you have seen this scheme before in multiple different iterations, but basically what the idea is you're starting with many compounds, thousands, tens of thousands of compounds, and you're going through a series of different screening and optimization steps with the hope that you are going to enter into preclinical development, your animal stages, and then into the clinical development with some candidates that are going to be both safe and effective. And then, of course, what the goal is to get one drug to market that's going to be successful and make your company a lot of money. The thing about this scheme is it's not a linear process. It actually has a lot of weighting as far as the time and money towards the end of this paradigm. Once you start to get into the clinical development, this is where you need kilogram amounts of the drug, hundreds of millions of dollars. This is years in the making. And so most of the cost to making a new drug is actually in the latter phases. At the beginning, the early phases of drug discovery, where you have a high amount of compounds, this is actually where the technology, the high throughput screening technologies have really had an impact, where you can go through these initial steps once you have a disease and a drug target, some target that is druggable with a small molecule. Then you can very rapidly take those tens of thousands of compounds, limit it to those that are able to interact with your target. Then you can start to prioritize as far as which of these hits become your leads. And then it's a process of trying to prioritize, move the safer products, the more effective products to the top of the list before you get into these more expensive processes. So on paper, this looks like a very good idea where you can screen as many potential drug candidates as you can to get to that ultimate effective drug. But in fact, there has been some problems with this approach. First of all, the rate of success is quite low. So what's shown down here at the bottom is just a graph of what percentage of the drug discovery programs actually result in a drug hitting the market. And it's somewhere around 10%. So all of the resources that went up to getting that drug ready for the preclinical, the clinical development is being wasted because you're not going to recoup them from selling a drug into the market. What are some of the reasons for failure? Well, it could be that once you move into the preclinical and the clinical phase, even though they are showing activity in the early phase, they're not having efficacy. And also a big issue that we'll talk about today is that they are deemed to be unsafe, or they're having adverse events once you get into these in vivo stages of drug discovery. So some other concepts that I'd like to discuss that pertain to how you use these screening methods are shown beginning with what is a pharmacophore. And this is really trying to describe your targets or your leads and how they are interacting with this main drug target, which is generally a macromolecule uh, of some sort, a protein, a receptor, an enzyme, or even DNA. And so a pharmacophore is describing this bimolecular interaction between your drug and your target. And this really has to do with the different steric and electronic and structural features of both constituents and how they are interacting with each other. And whether this interaction has sufficient energy, sufficient conformation in order to lead to a biological effect. Quite often people think of this as a lock and key mechanism, where if your key or your drug is fitting into the lock, your macromolecule, with the appropriate conformation, then you get an on state. So some of the types of interactions that we're talking about here, hydrogen bonds, hydrophobic areas, different charge interactions, which allow for this enabling interaction. For a interaction that might be consistent with an antagonist, it may be able to interact with this key, but there are bonding interactions that are repulsive in nature, and so you don't get an effect. Unfortunately, as we'll talk about, this is an oversimplification. This may describe binding and affinity, but it doesn't explain the activity of this liganded complex. So again, trying to think about how we move into this hit to lead part. So we have a druggable target, and we may have a hit or a reference compound or some sort of a scaffold that we now want to utilize in order to build some related pharmacophore molecules that we are predicting are going to interact with our drug target. And so there are mechanisms out there now for high throughput synthesis, taking a scaffold and adding to it. There's computational design that can be used to try to optimize this fit. And then we can use high throughput screening mechanisms in order to look at these hundreds and thousands and even tens of thousands of chemicals and say, is it interacting appropriately with our drug target? Now, one thing to think about is this is kind of a two-way structure activity relationship. You have to consider not only the structure of the drug, but also the structure of the receptor or the macromolecule. So what I'm showing here is a phylogenetic tree of the nuclear receptor family. And branches of this tree that are more physically close to each other, it means they're more related. They have more structural relationships. So when you're talking about your interaction of your drug with this ligand binding pocket of your drug target, you have to think about, well, what is the structure of that binding pocket? And then this idea of polypharmacology is really that no chemical is going to have only one target. There are multiple possibilities for what this drug is going to interact with within the cell. What tends to happen is that this particular molecule, if it's interacting with one receptor, it may have a higher tendency to interact with receptors that are phylogenetically related. So this is something that we'll describe later on as we're looking for the difference between an on-target, such as in this case, interacting with our on-target ROR, versus off-target, any interaction that we are not anticipating or we would prefer not to have. The other thing that we have to appreciate is the fact that not all binding events are the same. That even though they associate, these two macromolecules associate with each other, there can be differences in what the ultimate effect is. So when we're starting to look at the screening of our on-target and our off-target effects, we have to think about several parameters that we can use for comparison. Things like affinity, how strong does our drug bind to our receptor? How much activity are we seeing, so the efficacy? And what dose do we see a certain level of effect or potency? And there are all different measures for this, but the idea here is that we have to be able to quantify what the effect is for a particular interaction so that we can start to prioritize its importance in the drug discovery scheme. The other thing to keep in mind is that the measure of interaction of your drug with a particular target, no matter how much quantification we have, is not enough to define what is the best drug. This is simply just looking at the interaction between one drug and one target. So we have to start thinking about, well, how do we define any other effects of this particular drug on the cell? So in summary, when we're looking at the classic drug discovery paradigm, we talked about it's a high cost, low success rates. We have the problem with drug promiscuity, where we have multiple off-target possibilities for our chemical. Activity is not as simple as on and off. We actually have different levels of activity that we can see. A couple of things that I didn't mention is that as we move into the preclinical phases, quite often our animal studies are not predictive of the human response. So we need a better approach, or we need to add to this current approach so that we're able to make better decisions about which drugs to move forward into our clinical trials. So this is largely described as drug discovery toxicology as kind of the addendum to classic drug discovery. So what the idea here is to screen for known toxicity pathways early in drug discovery where it's cheap and where the methods are higher throughput. We want to prioritize those that we predict to have a higher safety profile. We want to keep in mind that all drugs are going to have some level of off-target effects. So this idea of polypharmacology, multiple pharmacological interactions, and both the chemical and the protein are important as far as structure-activity relationship. And also, again, if our ultimate target for the clinical setting is humans, then we're going to want to make sure that we're doing as much as possible in a human situation as we can, mainly looking at human drug targets. So the discovery toxicology aspect is really, again, just an addendum to what we've been looking at for classic drug discovery. And we're going to divide this into multiple types of screening that are done. Prospective screening, where we're looking at off-target liabilities for specific subsets. Signal generation screening, where we're trying to look at multiple liabilities. And then retrospective screening, where we're taking information from later in the process and trying to move it into the early phases of drug discovery. Just very quickly, we'll just describe nuclear receptors. Of course, you can find a lot more information on the tab section of this webinar, but also you can go to the Indigo Biosciences website and go to resources there to learn more about nuclear receptors. I just want to cover a couple of basic features about nuclear receptors. First, this is a family of related proteins. There are 48 different nuclear receptors in humans, and they all share a similar structure as well as similar mechanism of how they change cell fate. So the general structure is shown up here, looking at the primary sequence, and then down below is showing some secondary sequence of features of this family. So all members of the nuclear receptor family have this same type of DNA binding domain. So they're able to interact with specific sequences of DNA And they may do this with a homodimer, heterodimer, or as a monomer. And there's also a conserved region that is the ligand binding domain. And again, this is conserved among all 48 members of the family of nuclear receptors in humans, but they vary enough in both the DNA and the ligand binding domain where you get a different biological niche for each receptor. The key aspect of nuclear receptors for the screening assays that we're going to describe is that these assays are ligand activated transcription factors. And so they're interacting with a piece of DNA for genes that they're going to regulate. When a ligand binds, it will cause a conformational change that is then going to recruit the coactivator complex, a transcriptional machinery, which is then going to drive the transcription of a certain subset of genes that are responsible for the effect of each individual receptor. Now, again, I mentioned this before, in that binding is not enough. It's really the conformation of the liganded complex that translates into cellular activity. And nuclear receptors have a very interesting, diverse way in which ligands can change activity of their target protein. And what's shown in this cartoon is the unliganded binding pocket. In this case, this is showing the estrogen receptor, where all of these conserved regions of the ligand binding domain are folded in a certain way. Then when a ligand interacts in a permissive manner, this causes a change in shape in the protein, which then able to recruit these coactivator complex that leads to an agonist response. If you have a compound that is interacting in a non-permissive fashion, this interaction causes a different conformational shape or a non-permissive shape where you no longer get transcriptional activity. And then again, this isn't a switch type of model. You can actually have an in-between effect, something in between a full agonist and a full antagonist. And this is called a partial agonist. And this is where the conformation is leading to some interactions within that binding pocket, but it's not leading to a full permissive structure of that liganded complex. And that's shown in detail in this dose response curve on the right. Full agonists lead to 100% activity within that cell system. Partial agonists, even at high concentrations, are not able to reach that full activity, again, because the shape is different. A full antagonist is non-permissive, so you don't see an effect by the antagonist by itself. But note that this is interacting with the binding pocket and not allowing the agonist to interact. So in the presence of an agonist, an antagonist will lead to this decrease in response that's shown on this bottom graph. Now, nuclear receptors have another type of agonist where this interaction within the ligand binding domain causes a conformational change that recruits transcriptional repressors. And this type of a ligand is called an inverse agonist. So again, ligand just means it's binding, but you can have a multitude of different types of activities depending on how that ligand is binding to the target. So in the drug discovery space, nuclear receptors have a lot of importance. First of all, these are on targets. So roughly 15 to 20% of all prescription drugs will target a nuclear receptor as an on target. Things like estrogen receptor being targeted with tamoxifen, fibrate drugs interacting with PPAR alpha. Importantly, and this is something that we'll talk about a little later on, is that these are important off targets as well. So there are several nuclear receptors that are involved in adverse events that we want to try to limit the interactions with these particular receptors. These are also targets for different nutrients and as well as microbial products. So there's a lot of different endogenous and exogenous ligands that can be important as we're going through and prioritizing our drug targets. So when we talk about screening in the future slides, what we're actually talking about is using some cell-based assays where we can specifically look at each of these different nuclear receptors. Again, this is something that you can get more information on the Indigo Biosciences website. But what we're using is engineered cells, where we are tricking those cells into responding to a ligand. Instead of regulating an endogenous gene, we are having it regulate a reporter gene. In our case, we use luciferase. So we can interrogate a wide variety of different nuclear receptors and look for their ability to be activated. We can look at a variety of different transcription factors. We can devise different types of cell systems that will be very specific for receptors that are closely related to each other. So we have a very robust and specific way that we can address whether a chemical is interacting with a nuclear receptor. Okay, so now that we've talked about the drug discovery process and some basics of nuclear receptors, let's go in and talk about how we can utilize our knowledge of these approaches to try to find better drug candidates. So let's start with what's called a prospective screen, or looking forward. So these are predictive screens, and they're often called triage assays, where we are trying to remove compounds that may ultimately lead to an adverse event. So in our case, what we're trying to do is utilize what's called an adverse outcome pathway, where we are using pathways that have been developed through a lot of research into how drugs cause their negative effects, defining their mechanisms, and defining ways that we can predict whether a compound is going to have an adverse event. So what's shown down here is a simple linked pathway of how a drug or a xenobiotic ultimately leads to an adverse outcome. So in this case, what we're looking at are linked events, like a molecular initiating event. This is an interaction with the drug with some macromolecule. That macromolecule leads to an effect within the cell, which leads to a secondary response, which leads to an organ response, and then an overall toxic response. So these have been defined. We know how each of these events are related to each other. And importantly, we can use this as a way to screen compounds. So we can ask a simple question. Does my drug interact with this molecular initiating event? If it does, because we know this linkages, this is predictive of an adverse outcome. So this is being used quite a bit in the drug discovery space currently, looking at drugs that are going to have effects on cardiac function. For example, if your drug interacts with a calcium channel, hERG, then it is associated with causing arrhythmias. If you have compounds that cause DNA damage, something that we can easily screen for using something like the Ames assay, then this is predictive of mutagenesis, could be teratogenicity or cancer. There are several adverse outcomes where nuclear receptors are the molecular initiating events. I'll describe one of these in the next slide that has to do with drug-drug interactions, where certain nuclear receptors are regulating the pharmacokinetics of other drugs and other nutrients. But we also have to be cognizant of the fact that these nuclear receptors have a very important endogenous role in things like steroid metabolism, in endocrine activity, androgen activity. So anything that's interfering with the activity of these steroid hormone receptors can lead to endocrine disruption, can lead to reproductive effects, can lead to increasing risk of cancer. There are several other adverse outcomes that have to do with liver pathology, things like steatosis and cholestasis, where there are key players within the nuclear receptor family. As I mentioned, this idea of a prospective screen to try to get rid of drugs that may potentially cause drug-drug interactions is something very important in the nuclear receptor space. In particular, there is a receptor called PXR, which stands for pregnane X receptor, that is a xenobiotic sensing receptor that's found in high concentrations in the liver. And its primary role is to increase the drug metabolism capability of the liver to try to get rid of these xenobiotics. So it regulates phase 1, phase 2, and phase 3 drug metabolism enzymes, things like cytochrome P453A4. And this is a very important enzyme for increasing the turnover of roughly 50% of all prescription drugs. So this concept of drug-drug interactions is that interacting with one drug interacting with the nuclear receptor is affecting the absorption, distribution, and metabolism of the victim drug or the drug that is interacting. So we can easily screen for some of these potential compounds that are going to cause drug-drug interactions. What's shown over here is one of our most popular assays is looking at PXR activation. A variety of different drugs, a huge variety of different structures represented here, and they're able to activate PXR. And again, this would be predictive of a potential drug-drug interaction. And a triage assay is something that we can get rid of. So the idea here is we can take these hundreds of chemicals, use our high throughput screening capabilities, and remove those that have a liability. In our case, we may want to remove drugs that would cause drug-drug interactions, endocrine disruption, steatosis, etc., before we move them into any other optimization. The next step is what's often called signal generation. This is also called safety pharmacology, where we're trying to understand how specific your compounds are. Are there off-target liabilities? What is the dose response of these off-target liabilities? And then taking a look at what is the biological niche that's associated with some of these off-targets. How concerning are they to the potential success of your candidates? These utilize, again, high throughput systems, cell-based assays. We can utilize the human cells or human targets to try to get to the ultimate clinical studies. These can be targeted, so we can look at a protein family or subfamily, or we can look at everything that we have, just trying to cast as wide of a net as possible. The two major types of signal generation screening are omics, using omics technologies to look at a wide range of different kind of descriptive effects that happen when a chemical interacts with a cell, or transcription factor profiling. So something like transcriptomics is a signal generation assay. You treat a particular cell model with your compounds, and you ask the question, what are the effects that are seen? Are there some adverse outcomes? Are there some changes in gene expression that are predictive of an adverse outcome? Transcription factor profiling is looking at the actual molecular initiating event. Can be very high throughput and can be very specific. So to try to differentiate between the two, as we're looking at this adverse outcome pathway, something like transcriptomics is actually looking at the key events that are associated with an adverse outcome. Transcription factor profiling is actually looking at the molecular initiating event. So what we're trying to do here, this is a major prioritization step where we may have an idea of what our priorities are when we have our leads, just looking at our on targets. We may have an order of potency, order of efficacy. But now as we start to overlay some of these off-target effects, some of these liabilities, we have to factor these in. There may be some off-targets that are akin to a triage assay where we remove drugs, or these may affect how we prioritize based on dose response, where these off targets are seen relative to the on target. The last part of this discovery process are called retrospective screens or repurposing screenings. And these are generally when the compound has moved along to the in vivo phases, so the animal studies, or into the clinical studies. And these quite often are not predicted. So there may be some effect that is seen at a much lower dose than we would have expected, or a completely unexpected adverse outcome, something that we would consider idiosyncratic. This may also have to do with a species difference as we're looking at effects in an animal model that we had not taken into account from our in vitro studies. So in order to take these retrospective questions and move them into the drug discovery process, we want to gather as much information as possible about what is the type of response, when does it occur, at what dose do these events occur at, is there some structure activity with my candidates that can help explain this. And really what we're trying to do is come up with a potential molecular initiating event that resulted in this unexpected adverse outcome. And then if we can determine or hypothesize a molecular initiating event, then we can develop a screen in order to eliminate this in the future, but we can also start to better understand what the liabilities are of some of our candidates. As I mentioned, species differences is something that is always a difficulty in the drug discovery process. And this is true with the nuclear receptors as well as with other types of molecular initiating events. So actually several of these molecular initiating events have very distinct species difference. What I'm showing here is PXR, and you can see that some ligands like this rifampicin, the antibiotic, has no effect on mouse and has a very nice dose response with the human. So this is telling you that the mouse may not be the best model in order to look at what the effect of rifampicin might be on drug-drug interactions, for example. The other thing that we can do is we realize that there are some molecular initiating events that we look at that are not pertinent to human health. For example, PPAR alpha dependent activation in rodents leads to tumors in the liver. That is not of human relevance. That does not occur in humans. Another classic example is drugs that bind to alpha-2U microglobulin causes kidney toxicity. Well, this is a pheromone that's only expressed in rodents. So obviously this type of toxicity is not important to humans. So again, we can use this information in a retrospective manner to try to make better sense of our clinical and preclinical studies. Now, this brings me to something that is also a retrospective screen that has to do with repurposing and going back to this idea of polypharmacology. So this is where one drug has multiple targets that it interacts with. And we've talked about this solely as a negative aspect, a liability. Well, there are ways of actually kind of spinning this a little bit. And we can actually now repurpose drugs that were developed for one aspect. And now since it has a different off-target, different pharmacology, we might be able to target a different disease. So what's shown here is, let's say that we have our drug that interacts with our on-target with ROR gamma, which is involved in inflammation. And let's say that's made it through the clinical trials. Everything looks great. We also look at our drug and realize that when we did our retrospective, when we did our safety assessment, safety profiling, it also had anti-androgenic activity. Now we know that anti-androgen drugs are important ways to treat prostate cancer. So now this one drug we can repurpose. It's already made it through the FDA process. We know it's safety profile, but now we are repurposing it for another endpoint. And so this, again, utilizing some of our information that we've gathered throughout this process, but we're now repurposed it back into clinical development. But other aspects of this retrospective screening, again, can inform the beginning part of the process, where we can reprioritize our drug targets based on what we learned from our preclinical studies. And we can actually develop prospective screens where we can eliminate any drug that is going to cause us problem later on that we hadn't anticipated in our first round through this paradigm. Okay, so now that we've kind of gone over the concepts, I want to just quickly go through an example of how clients interact with Indigo to try to increase the information they have on their drug leads in order to move into their preclinical studies. So in this case, the client came to us and they had an on-target. They had several drugs that they felt were going to be targets for a target called ROR gamma, which stands for RAR orphan receptor, very important in inflammation and autoimmune disease. And they had a series of compounds. I'm just showing you the first 17 where they wanted us to screen for activity. ROR gamma is one of those receptors that exhibits inverse agonism. So this is a decrease in activity. I've kind of flipped the dose response curve to make it easier to look at. We screened the compounds and compared everything to the reference compound for this assay. You can see here a variety of different dose response curves. We can look at the affinity, we can look at the efficacy, and we can start to rank some of these drugs as far as their ability to interact with the target. Bring this question up again that I showed in an earlier slide. What makes the best drug, which will have the least harm? You cannot get that information from this simple preliminary screen. The next step is a triage screen. And so we took these compounds and we said, well, we're concerned with PXR activation, its ability to lead to drug-drug interactions. This is something that the FDA is asking of the pharmaceutical industry to examine. We looked at these 17 compounds within their portfolio, or client's portfolio, and simply asked, are we getting activation of PXR, and at what dose are we getting? You can see here that we did have a couple of compounds that were able to activate PXR, tended to be shifted relative to the on target, but nonetheless, this was an activation. We know the affinity. This is information that we can move forward. The client can decide that we want to remove these compounds, or at least they know that information so that if this does become a drug, that they may want to label it as a potential liability for drug-drug interaction, and they can even state what other pharmaceuticals might interact with their new drug. Now, selectivity screening is part of the safety pharmacology. Closely related member of related to ROR gamma is ROR alpha. Very similar in a lot of its ligands. But as you can see here, there were some compounds that showed some activity towards ROR alpha, but many of them didn't show much activity. So there was some selectivity associated. Okay. Now, again, this is a place where the client has to decide how important is selectivity. Do they want a compound that only activates ROR gamma? Or will they allow for other closely related members to interact and have activity? And I put in this comment about Vioxx. Vioxx was a specific cyclooxygenase 2 inhibitor that ended up having to be removed from the market because it was so selective for COX-2 that there was a rebound effect due to a closely related member called COX-1 not being inhibited and trying to compensate for that loss in COX-2 activity. And so it resulted in some cardiovascular effects. And then finally, the kitchen sink approach. Let's try to look at as many liabilities as we can. So the client may ask us to look at every single lead compound in every assay you have, regardless of whether it's a nuclear receptor or not, in every mode. So agonist, inverse agonist, antagonist mode. We may start with just a single concentration, and then if there is activity, then go into a dose response. But again, we are trying to cast a wide net. We're trying to understand any potential liabilities that are here. This is showing a heat map. Anything with a red color, more red color means more activity. You can start to sort. I'm just showing you the agonist activity here, looking at all the different assays that were examined, and then all the different compounds. And you can start to compare activity. You can use this hierarchical cluster as I did here on the left. You can use these spider plots to try to look at what are some of the activities. Here's our on-target activity. Everything else in this case would be off-target. And then you can say, out of these compounds, what spectrum of responses are most important to me? Again, similar to specificity, but which off-target effects concern me, which ones may actually be necessary or wanted. So this is something the client needs to decide, but they have all this data now to examine it. So one thing that I'd like to point out from this kitchen sink approach is that you can start to see some activities that maybe you had no idea that there was structure activity relationships towards a particular other molecular initiating event. So this leads to that idea of repurposing. And I've highlighted this slat here that has to do with the agonism of PPAR gamma. And you can see that this compounds 5 and 6 had very good activity in a single dose. We went back into dose response. Sure enough, these compounds have PPAR gamma activity. So again, is this a concern or is this a benefit to you? At least you know that information, and now you can prioritize. So in conclusion, we talked about some of the problems with the current approach to drug discovery, the cost, the time, the low success rate. So anything that you can add to improve your knowledge of your drugs is going to help you have a beneficial outcome. So things like prospective screens, looking forward, trying to triage compounds, signal generation, looking at selectivity, looking at potential off-target effects, and then retrospective screens, looking backwards and trying to utilize mechanistic information from your in vivo studies. All of these things add together to look at your liabilities, look at your potential for leading to a successful drug discovery process. Through all of this, we've utilized as much molecular information as we can, predictive assays such as those that are molecular initiating events within adverse outcome pathways. We are utilizing as much of the information as we can about each compound as far as potency and efficacy towards multiple targets, how selective it is, this idea of polypharmacology, are there multiple targets? And again, all of these things are meant to make the decision process not only cheaper and faster, but in the end, you're going to have safer drugs that are making it into the market. So with that, I'd like to thank you for your attention, and I am available to answer any questions you may have.
Fantastic. Well, thank you, Jack, for a really great presentation here. Alrighty. So we'll start off with a great question here. So how do in silico methods complement or compete with this type of cell-based screening?
I would say they definitely complement. They don't compete, at least not at this stage. So the in silico methods, things like computational models, docking models, some machine learning types of approaches, all of these I think have applications for trying to narrow down the types of chemicals that are interacting with a particular target. It's particularly useful when you're looking at a single target and a wide variety of different chemicals. So in that regard, I think they help streamline the drug discovery process. Where the information is kind of lacking, or where I have less confidence, is thinking this some of these in silico models as being a prospective screen. So if you have a compound and you're asking the question, does this interact with a particular target, some unknown chemical, and just using purely in silico methods, I would say that the predictability is not high. So there has been some work done on specific pharmacophores, and roughly anywhere from 10% to 70% of the compounds that were predicted to interact using in silico and docking methods actually turned out to interact with that particular target.
Fantastic, great answer. Another good question here. So what are the advantages of a drug with multiple targets versus given two drugs, each with their own target?
Yeah, I like that question. That is very interesting. So, if you're talking one drug, and let's say it has two different targets that it interacts with, versus taking two drugs that each interact with those same targets. So you may be able to play with some of the doses to hit those two targets if you have two different drugs. But then you also have to counter that with the fact that you have two different drugs that you're giving with different pharmacokinetics, different ability to get to the target site. So it becomes more of an issue of trying to manage once you get into the in vivo situation. Whereas if you have polypharmacology, it's one drug, one pharmacokinetics for that one drug, and then multiple effects.
Excellent. All right, and here's a nice timely question considering the changes to the FDA Modernization Act, but could these assays potentially eliminate animal or preclinical studies?
Boy, I wish, but I think that we're a little ways off from that. So what the idea is you certainly can decrease the number of animals that are required. We can screen for activity within animal models using animal targets instead of actually using the animals themselves. We can predict some adverse outcomes using these in vitro assays without having to go to an animal study, but we're not able to cover all the adverse outcomes. So cancer, for example, we can't screen for that very well right now using in vitro systems. Teratogenicity, some other fibrosis. These things are too complicated for us to be able to use our cell-based assays, use cell culture model systems. But hopefully over time, we're going to be able to get to that point.
Fantastic. We still have a bit of time, so it'd be good to ask a forward-looking question if you don't mind. How do you see the use of cell-based reporter assays and nuclear receptor profile changing in the next 5 or 10 years?
I think it's probably going to continue to advance as far as being able to add high throughput capabilities. It may also be improved through what's called precision medicine. So instead of looking at assuming everyone's estrogen receptor is the same, look at different genetic variation within the estrogen receptor, or even looking at mutations within a receptor and say, does this mutation have the same pharmacological profile as the wild type?
Yeah, great answer. All right. Well, Jack, thanks so much for your insights today. It's really been a pleasure having you with us.
Thank you.
Big thanks also to the audience here for participating. And in closing, we hope you enjoyed this Scientist.com Inside Scientific and Indigo Biosciences webinar. And we'll see you again next time. Have a great day, everyone.